Can we as humans access immortality? The simple answer is yes, and we know that other species can do it. We don't know their lifespan, and they can exist forever. In 1938, Hermann Joseph Muller and Barbara McClintock theorized about protective cap in top of the chromosomes working with the DNA of a fly named Drosophila melanogaster, who doesn't manifest alterations such as deletions or inversions. In the early 1970s, Soviet theorist Alexei Olovnikov first recognized that chromosomes could not completely replicate their ends. This is known as the end replication problem. Leonard Hayflick established his limit of cell division by 1984. Telomerase was discovered by Carol W. Greider, Elizabeth Blackburn, and Jack W. Szostak. Telomerase, also called terminal transferase, is a ribonucleoprotein that adds a species-dependent telomere repeat sequence to the 3-foot end of telomeres, which can reproduce the missing part of the telomere to protect the ends of the chromosome and ensure its redivision to produce another healthy long telomere chromosome. The taller the telomere, healthier is the cell. In the human body, there are 3 types of cells: somatic cells, who forms the body parts, and germinal cells or reproduction cells, eggs and sperm, and there are stem cells or undifferentiated cells. Germinal cells are immortal as they should be to not end up with old babies, and believe me that these exist, named progeria, which is a genetic mutation causing premature, elderly symptoms. Cancer cells are immortal because telomerase is overexpressed in the cells because of genetic mutation which lead to the immortality of the cancer cells. In 1951, a female cancer patient named Henrietta Lacks died and donated her tumor to science. The cancer cells, named HeLa cells, grow to be 50 million metric ton. In 2012, Jennifer Doudna and Emmanuel Charpentier re-engineered the Cas9 endonuclease into a more manageable 2-component system by fusing the 2 RNA molecules into a single guide RNA that, when combined with Cas9, could find and cut the DNA target specified by the guide RNA. This contribution was so significant that it was recognized by the Nobel Prize in Chemistry in 2020. So with the discovery of CRISPR-Cas, we can genetically edit the part of the DNA that we want to express it or not. What we want in the cancer cells is not to express the telomerase to make it mortal, but for the rest of the healthy body we should do the exact opposite, but to express as much telomerase to fix the damage occurred in the end of the chromosome to make it divide properly and undefinitely. But the problem is how to put desired Cas9 or CRISPR-associated protein 9, the DNA fraction that we want, turn on or off into the body cells one by one. To avoid the problems related with inert viruses inserted to the body, there are a molecule named morocalcine. It's a protein, 33 amino acid residues in length, isolated from the venom of the scorpion Morus palmatus, which belongs to the family Chactidae, first characterized in 2000, which have the properties of cells' membrane permeability and can hold any molecule of any size and deliver it in to the targeted intracellular location to fix the genetic problems. Other problems who come with aging is body cells loss. In 2012, Dr. Shinya Yamanaka has the Nobel Prize for his discovery of the induced pluripotent stem cell, why can transform somatic cells with specific genes into pluripotent, then totipotent stem cells to redirect them to fix any part of the human body, so we can with blood cells have a bank of cells to fix our body when we need to. This genetic engineering methods can be effective against other health issues like atherosclerosis, hypertension, familial hypercholesterolemia, and many others.